Award during his postdoc and got a prizes for his doctoral studies as well. He's got a large amount of grant funding, and what his lab has been interested in is understanding how cells organize to form multicellular structures and to coordinate how they behave. And he's going to tell us about one aspect of this in this talk about cell cooperation in the tumor microenvironment. So Carlos, welcome, and good to see you back again, or see you again, I mean live, and the floor is yours. Great. Let me just give you this bit.
Thank you, and for the invitation. It's been very fun talking with people here. Had a pretty cool lunch with the trainees. So I'm gonna discuss some of the stuff that we were talking actually over lunch. And I'm actually gonna talk about maybe two stories. So the first one is gonna be like a short project. The project is not short, unfortunately, but it's going to be like the first part of the talk is going to be a little shorter, but I think it's going to give you an idea of the type of questions we're interested in. And then I'm going to try to dive a little bit deeper into one specific project. I like to say, as most of the work of my lab, this is unpublished. So I think this should be fun. So let me start with this picture here. This is a photo of Sao Paulo, actually. I was talking with some Brazilians here. And this is actually very common everywhere in the world, South America, as an example. And you can clearly see that there is very clear differences in the size and the types of buildings you have, probably due to different access to resources, right? And that differential sort of resource accessibility, it not only determines whether the type of housing you live, but probably also affects the education level you get to, the perspective of your kids going to college and so on, right? So there is these structural problems where access to resources can have a big impact in this case, people. So I want to try to think about that a little bit in the context of cells and in the tumor. And I wanted to start talking about cell atlases, which is sort of been a big boom in cancer biology, especially with the sort of single cell sequencing technologies. And I am old enough to actually have used atlases when I was a kid. And to me, one of the things that is missing from these single cell sequencing atlases, and I know many of you are actually working hard in improving this, is that it really seems to be like a list of different cell types and different phenotypes that you get. So that's kind of like the most boring part of the atlas, which was the index at the end where you can find the different locations, right? But to me, the sort of maps, the location where things are find themselves, it's what it's really interesting, right? I was actually discussing this with the students and postdocs during lunch. In developmental biology, people have been studying these cells and how they differentiate in 3D and in their sort of time and space forever. And now that single cell sequencing technology came in, it was like a dream technique for a sort of whole landscape and set of coordinate and frameworks that they already had and they knew existed. In cancer biology, though, I think it seems that we sort of got these new technologies of single cell sequencing, but we don't really have clear ideas of what are the axes that determine this heterogeneity, right? And again, I know people are working hard on this, and the advent of spatial transcriptomics and other omics is one example where this is getting really, really exciting. So how are maps drawn? And there are some obvious conditions that, for example, can be the natural environment. So again, Brazil, I'm picking on Brazil for no reason. This was before I met you today. So in this case, you have three countries separated by a river, right? So that's a natural environment separating this. But then obviously humans have a big impact. So this is a border crossing between China and Mongolia, Poland and Ukraine. And obviously sort of social and societal conflicts and sort of forces also determine borders. So this is some enclaves here. You can see in it's like really a mess. And then you can see also the opposite in Africa, where, you know, some people just like draw lines and decided these are countries. So all this is to say that what determines the borders can be multifactorial, but it's crucially important if we want to understand how, in this case, would be countries or sort of these regions operate, right? So, funny enough, we are in the geology room. So, in a way, geography is the study of how people or countries and separate in space. And part of the interest of my lab is it's maybe a sort of tumor geography, right? So how are these different cell types distributed in tissues or in tumors, and why are they specifically located in those locations, and how that affects their behavior, right? So we try to address this problem through different angles. So the first part I wanted to talk to you, it's about sort of tissue heterogeneity and sort of pattern formation within the tumor. So just like resources, access to resource plays a big role, and perhaps in cities, in the tumor, the proliferation rate of solid tumors is way faster than their ability to make new blood vessels, right? So what you can see here stained in brown are blood vessels in the periphery, in the sort of peritumoral tissue. And hopefully you can see that there is a lot of vessels per cell nuclei, whereas in the tumor, you don't have enough blood vessels for every cell, right? And again, that's sort of an intrinsic problem of a fast proliferating tissue. And my background in UCL, actually in the UK, was developmental biology, where it's all about how gradients of signals determine different tissues and specify different cell types, even if those cells may be genomically identical. So a big part of the lab is trying to understand how the distance to a blood vessel may provide and create different metabolically different environments. For example, the levels of oxygen, the level of nutrients, and how those different metabolic conditions and environments may dictate different cell phenotypes or cell behavior within the tumor. There's obviously a big contribution for clonal heterogeneity and mutations, but even on top of that, you have these sort of non-genetic sources of heterogeneity. And this is particularly important for stromal cells where mutation burden is actually much lower, right? So this is immunofluorescence of a tumor section, just to show this is not just on my mind. So I'm staining here in white for a probe that labels hypoxic regions. And in red, you see an immunofluorescence detecting blood vessels. And I actually was talking with Gilbert this morning, and he has like some really cool images showing a very similar thing. But what you hopefully can see here is that this region of hypoxia are sort of mirroring the topology of these blood vessels, right? So as expected, it's not randomly distributed. There is this sort of almost like unidimensional gradient, in this case of oxygen, and you can quantify that. And the question is, how is that affecting cell behavior? So, or cell phenotypes, right? So one way we study this is by looking at tumors, right? So you can image an entire tumor and then find individual cells and quantify different markers. And then you can do some sort of cell cytometry, right? So analysis of proteins. This is, you guys are doing even much fancier things, like with either mRNA and multiplex immunostainings, and also these pool guides. Sounds really cool, but the idea is the same, right? You can basically use microscopy in large areas with high resolution, so you can get sort of spatial information together with whichever omic or markers are you staining for. So this work, I'm sure you all read it because it's published. So, but we basically found that at that time, people were thinking that tumor-associated macrophages were co-expressing these two markers. And what we showed was actually there were very distinct subpopulations of macrophages. And one in particular that is particularly bad for prognosis was almost exclusively located in nutrient-deprived regions of the tumor. So to test that, we developed this, I call it a simple system with a fancy name, but it's basically an ex vivo system where we can culture any cell that we want and expose them to the same type of gradients that you see in tumors. So the way it works is really simple. You basically put cells here, And the trick is that they're sandwiched between cover slips, but there is a little opening in one side. So that means that if you're a lucky cell that happens to be near the opening, you have full supply of oxygen and nutrients. But as you move in this direction, your neighbors before you start eating the nutrients and consuming the oxygen. So in this case, I'm just showing a tumor cell line that expresses GFP under hypoxia. So green means low oxygen. And they are cultured in this system. And if you zoom out, you can see this sort of really nice change across the gradient, right? So again, we're trying to have this sort of simple unidimensional, gradually changing environment, so we can try to study how cells change in these environments. So remember, I showed you that in the tumor, we found this arginase-1 positive subpopulation of macrophages only associated with ischemic tumor regions. And we can recapitulate here the same thing, right? So we can co-culture macrophages with tumor cells. These macrophages are stained with CD68. They almost never express arginase-1. You see one there, here, and another one there, and another one there. But as you move into these more nutrient-deprived regions, you see that virtually 100% of the macrophages switch to this specific phenotype. And at that time, people were surprised, so surprised that it took us a while to get it accepted, because most people were really focusing only on chemokines modulating immune phenotypes. And now it's much more accepted, and that metabolites do this as well. But I think this was one of the relatively early evidence for this. So you can also study this in 3D, but what I wanted to show you just very briefly is that this goes beyond just tumor cells and macrophages, and this is something that we are working on, and a very talented student is focusing, which is what happens with T cells and tumor cells, right? And many of you may know that immunotherapy has been very exciting, but it does seem to be much more efficient in liquid tumors and sort of leukemia than in solid tumors, especially tumors like pancreatic ductal adenocarcinoma that happen to be very, very ischemic. So this is a very simple and widely used system where you have some melanoma cells that have an endogenous peptide. The T cells, a specific type of T cells from a specific mouse, detect and kill the tumor very rapidly. So you see the pink is like a non-viability marker, and within 24 hours, the entire tumor is killed. So we can recapitulate these in our little devices. And what you see is what you, in the front is what you expect. So basically, every tumor cell gets killed within 24 hours. As you move into the more ischemic regions, there is virtually no tumor killing. And we can show that those T cells are completely viable. If you take them out, they can start killing again. So there's something about that environment that prevents them from killing. So we're hoping that this is, maybe one of the reasons why immunotherapy is failing. Now, I love microscopy. I hope you too. But in a way, it's limiting, right? So you can look for very specific markers, and I guess with some of the techniques many of you are using now, you can start doing more unbiased microscopy. But what we wanted to do is to adapt this system to make it more compatible with any kind of omics. And me being sort of relatively naive, what I thought is like, why don't we put these cells in a tray? We can slide it into this modified version of this system, and then we can have this tray pre-cut in such a way that then we can split these regions into different sectors and do any sort of biochemistry analysis separate. And obviously, the implementation was a little bit more complicated than that, but it's working really well. So you can have different numbers of sections. We usually use 3, but in theory, you could do more, right? So just as a proof of principle, this is HIF-1 alpha. It's a transcription factor that is usually degraded, so you don't see it in cells. But when cells are hypoxic, it becomes stable, right? So you see it. And this is what you see here, right? So if you take cell lysate from these different regions, you see this very nice sort of gradient of HIF-1 alpha. So the system is working really well, and we've done now a bunch of metabolomics and so it works really well. But the one thing I wanted to show you today is that, which is also helps to validate that the system is working really well and it's related to this T cell killing, is that using sort of barcoded multiplexing, we can have in the same sequencing pool, right? We can have the tumor cells alone or the T cells alone on how do they look like when they are trying to kill each other in co-culture, right? And this is in normal conditions. And then we can study how that co-culture changes across the different environments. And hopefully you can see that in the more, like the better nurtured environment looks very much like the regular co-culture. But in the more ischemic regions, you start seeing new populations of tumor cells and new population of these T cells in the context of the co-culture and in ischemia. I'm going to skip a lot of data, but we basically found that, which to me is very interesting, a lot of the research in trying to understand how these metabolic conditions it's affecting immunotherapy, it's focusing on maybe the T cells are getting exhausted or deactivated. But what we found, it was really the tumor themselves were changing. And one of the main changes was that they do not really express or dramatically decrease MHC1. So for the people that are not immunology experts, and I'm definitely not an expert, MHC1 is this sort of protein tray that every cell has to take an exposed antigen with, right? So the way a T cell would recognize a bacteria or a virally infected cell, or I guess a bacterially or a virally infected cell, is it would recognize an antigen, but that doesn't recognize that antigen alone. It recognizes it into this specific protein that presents the antigen. So tumor cells that do not have MHC1 could be right next to, could have the antigen, but the T cell that recognize that antigen will not see it because it's not presented in MHC1. So that was very interesting for us. If you look in vivo, you actually see a very similar thing where tumor regions that are higher in MHC1 seem to be more well perfused, whereas more ischemic regions and hypoxic regions of the tumor seem to have lower levels of MHC1. And this is in mouse, but now we're working with a group in Canada trying to get clinical validation of hopefully validation of these. So this was very striking because you can have tumor cells now alone. And if you treat them with interferon gamma, which is a typical T cell signal, they massively upregulate MHC1, right? So this is how it looks like. So with interferon gamma, these cells respond with tons of MHC1, but back in ischemia, they just don't, right? And you need to believe me, but there's tons of cells here. They just very little MHC1. We show that actually they are responding to interferon gamma. So we also make sure interferon gamma is absolutely everywhere. So it's not a matter of not sensing interferon gamma, it's a differential response. So with the single cell sequencing data, we did some pathway enrichment analysis, identify key pathways, and then we did an inhibitor screen. And we found we have a couple of candidates, but for example, inhibition of the, or reactivation of the Hippo pathway through this inhibitor completely rescue MHC1 in these distal regions. So hopefully what we are trying to get here is that if this indeed is one of the mechanisms by which solid tumors escape immunotherapy or immune detection, combining some sort of therapy with these MHC1 rescuing treatments could help. So this is, preliminary, and we now are trying to move to in vivo work and some more clinical data. But I sort of wanted to give you a flavor of the kind of things we can do with these tools. Yep. Yeah, so for these experiments, I'm just using tumor cells and T cells, but you can have triple cultures. You could also, in theory, take, sort of a tumor suspension and try to reconstitute the whole thing. I think the advantage of this tool is its simplicity and sort of you can really control the parameters. So you can ask very specific questions and then I think get usually good answers. The more components you add, it presumably is closer to in vivo, but also more difficult to determine. So I think there is like a trade-off there, right? So, and there is a little bit of tinkering that every new cell type that you add to the system, you need to do before, the number of cells that you seed and for how long. So you can, in theory, add as many cell types you want. In these experiments, yeah. Yeah, so we are not really working. So in terms of differentiation, what we've worked the most is with macrophages. So one of the projects we did, and I hope we're submitting that soon, but I didn't mention, but this arginase 1 phenotype that emerged in macrophages in these ischemic regions, arginase 1 is a key enzyme of the urea cycle. So for us, it was very interesting that you have these metabolic stimuli, right? Like low oxygen and low nutrients and maybe high pH, low pH, high hydrogen concentration. So, and the response of macrophages is a huge upregulation of a metabolic enzyme, right? So we thought there must be something on the metabolism of macrophages that is changing. So we did untargeted metabolomics in macrophages in these different regions, and we actually found a weird polyamine that suppresses T cell killing. So really interesting. So yeah, so in terms of the typical macrophage stereotypes of differentiation, we actually see that it doesn't really fit exactly the traditional one. So I don't think you would see that on T cells. But yeah, that would be one of the things we sort of plan to do at some point, is to take maybe earlier bone marrow cells or T cell precursor and see if you add the signals and see if you have different outcomes. Yeah. 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 No, that's a great question. So one thing I would say, though, is that what we're trying to model, right, it's usually when you get T cells into a tumor, assuming they can infiltrate, right, they've already been stimulated in the lymph node, right? So they come there already differentiated. And also, those are often a multi-day, maybe week-long process. Most of the things we're studying are maybe a day or two long, right? So what I think is that, for example, T cell exhaustion. Yeah, sorry. Oh yeah. So the question has been if we've been studying T cell differentiation across this gradient, and whether we see different sensitivities in different T cell subtypes to this MHC1. And I was just randomly ranting about different things, as you've heard me. So, but yeah, basically, I think it's a great question. But I think the timescale of our experiment, so I guess what I was going to try to mention is that we think T cell exhaustion, I don't know, I mean, maybe this is getting too much into the details, but I think it's a very real thing that happens over a week or so or more in tumors. Our experiments are much shorter than that. So we don't see a lot of evidence of exhaustion, but we see this massive immunoevasion change that we are really interested in. So depending on the question, you may need to adapt the timelines, and this specific system may be better for shorter timescales than longer. And also in culture becomes difficult, like you kept the cells there for 3 weeks. Is it really, so there is some caveats. But in the next 2 hours, what I wanted to discuss, it's now it's yes. Yeah. So the question is whether in these co-cultures we see significant contribution to cell migration. And the answer is that the way we design this system, basically the height, the distance between the two cover slip, it's fairly large. It's like around 700 micron. So that creates artificially shallow gradients. So in a tumor or in a tissue, going from full perfusion, the border of the capillary, to complete necrotic core, it's about 100 or 200 micron. Because our gradients are super shallow, we extend that to like 10, like a centimeter, 10 millimeter, like a third of an inch, right? So that allows us to look basically at better spatial resolution because we're zooming in. Basically, we're making a 10 cell diameter problem, a 100 cell diameter problem. That is very convenient for the specific questions we are studying. It is not very useful if you're trying to study migration because we calculated, assuming a random walk, it takes about a month. It would take about a month for a cell to go from one end to the other, right? So in our system, there is no real meaningful contribution of cell migration, but that doesn't mean in real or in tumors there would be that would be the case. So we are actually developing a much sort of steeper gradient version that is much shorter because we have some evidence that there is heme attraction towards nutrients. And that's the kind of question we want to address with that. So in this problem, cell migration is not that relevant. All right, so what I'm going to tell you now has nothing to do with what I just told you. So, I mean, it has something to do, but it's not going to be gradients for a moment. And it's really about what happens in these low nutrient regions, right? So, okay, there are gradients, but what happens in those sort of low poorly perfused regions? And the reason why I think it's interesting is that we know tumor cells are incredibly proliferative, right? So they need tons of nutrients. So it's a little bit of a paradox that tumor cells are highly needy for nutrients, and we know they consume a lot of nutrients, but also live in poor nutrient conditions. Partly they are low, the levels of nutrients are low because they are consuming it, but there is still something that didn't really fully fit this. And what we're going to try to convince you is that cell cooperation is a critical aspect of this problem. So basically, this is what I was just saying, right? So there is this sort of paradox where tumor cells need a lot of nutrients, and in particular, I'm going to focus on amino acids, but they live mostly in low amino acid regions, right? So how that happens. So since this is a very diverse crowd of dry and wet biologists, I wanted to ask who has done cell culture? Simon, have you ever cultured cells? No? Huh? Who hasn't cultured cells? Okay, who has cultured cells? Well, I guess the rest. Have you noticed that if you seed cells at a decent density, they're super happy, but if you mess up your dilution, all the cells will die, right? So that actually has been known for almost a century, but I think it's a little bit confusing why, right? And this is the experiment, right? So you have exactly the same conditions. You start with a relatively normal conditions, and then you have the cell growing. And but if you go 10 times lower than that, all the cells die, right? So the reason why I thought that was interesting is that, so this is actually how the data would look like, right? Is that it seems that there is a critical density under which the population is no longer viable. And, sort of being a biologist, that doesn't really fit with the typical idea of what we think about cell growing and tumor clones, where they're always fighting and competing for resources. Because, the idea of, for example, logistic growth is that basically the less the merrier, right? Because the amount of resources is fixed or limited to there's like an upper bound. And then the less individuals, then the more resources per capita. So the ideal density in a purely competitive scenario is 1. So then all the resources are for me. Now, in nature, despite the sort of idea of competition, the Darwinian idea, actually cooperation is very, very common. I always say that the most simple one is that any sexual organism needs a mate to reproduce, right? So that is a non-individual autonomous process. And the key thing about cooperation is that it needs some sort of density or proximity to actually operate, right? If you can imagine that if the next mate from a snail is 100 miles away, it's going to take a long time to find that mate, right? So there is this density. So when you look at the sort of growth dynamic of a cooperative population, right, sort of the ideal scenario, it's right at the middle, right? So I have an equation because I know I'm in a fairly advanced theoretical department, but also in case we have Gen Zs, I added emojis. So this is a very common model of cooperation in ecology. It's called the Ali effect, where it's very similar to logistic growth, but then you add this sort of minimal population size. And the ideal growth is at the middle, because if you go lower than that ideal, cooperation becomes really inefficient because it becomes harder to find individuals to cooperate with. And then if you go higher than that, now competition and sort of resources don't become limiting again, right? So if you take the cell data, and we've done this with tons and tons of cells, it really looks exactly what you would expect from a cooperative model, right? So the other reason why we thought this would be particularly interesting for tumors is that one of the first person that described this cooperation during evolution, I just show you this photo, is this Peter Kropotkin, this Russian anarchist, very interesting dude. But part of his observation were looking at cows and other animals, farm animals in Siberia, and like, I've never been there, but I know that winters are cold, so they would see all these animals, sort of huddling together to survive the winter. And another great example, right, is penguins, right? You've seen probably documentaries where to survive the winter, they have to really make these big huddles, right? So the message is that often, not always, but often in nature, you see cooperation becoming more important when the environment is tough. And since the tumor microenvironment is such a tough environment, we thought that this could be relevant. So to study this, we tried to determine the Ali threshold. And it's basically, you can take it as the minimum size of a population that is able to survive in a particular environment, right? So you could imagine that a single penguin in very cold regions would die or sleep. Whereas when the weather is nice, you can have a single penguin with no problem, right? And maybe that's the future of penguins, given the way things are going. But in theory, at least, right, is the temperature becomes colder and colder, you would need to have a bigger group of penguins to not freeze yourself, and that means a bigger Ali threshold. So what we did was took a bunch of tumor cell types, and we starved them of different nutrients, and hopefully you can see that, for example, glucose starvation or the levels of glucose didn't really affect this minimum population site. But when you start tuning down amino acids, you see this very clear change. And again, this is a little bit counterintuitive because it's saying that the least amino acids you have, the more you want to be, right? Which is weird because it means more competition for amino acids, right? So this was to us an indication this was something perhaps understudied. And this is the same idea, right? You can see here, you see the same cooperative shape according to the levels of glutamine in this case. You see this Ali threshold shifting according to the cell number. So how do amino acids become cooperative, right? And again, for the most part, amino acids come through, obviously, amino acid transporters, which is really a cell autonomous process. There's many groups that have worked on cancer cells, particularly gobbling entire proteins through macropinocytosis. But that's also, sorry, apologies, especially on Zoom, sorry. But that's also a cell autonomous process, right? So how, right? Well, between the world of amino acids and whole proteins, there are oligopeptides. And the way I see the tumor is this like sort of almost like the video clip of I'm a Survivor. So it's like a jungle, and then the tumor has to like cut all this vegetation, which is the extracellular matrix, to actually invade, right? And we study that, but that means there's tons and tons of proteins that are broken down. Some of them may be eaten by macropinocytosis, but it's not crazy to think that there's a lot of oligopeptides going around in the tumor microenvironment. In fact, if you look at publicly available data, we see that virtually all oligopeptides, in this case, we were looking at dipeptides, tend to be much higher within the tumor than in sort of adjacent healthy tissue. Here I'm just showing you for all the ones we could detect with glutamine. And there's this one, I guess, glutamyl glutamine, a little bit lower, but most of them really increased in the tumor microenvironment. So we actually use so we've tested this in many different combinations. So it's not about just this one dipeptide called alanyl glutamine, but we're going to use it as a model system, partly because I want my lab to survive. So we can buy roughly a gram of this for $7, and it would cost you $35,000 if you custom made that. So, all right. So now, so for people that are less familiar with cell culture, there is this thing that I don't fully understand, but even if glutamine is a non-essential amino acid, meaning our cells are able to synthesize it, for example, from glutamate, our cells hate, and it's basically impossible to grow most cells without glutamine, without an external source of glutamine, right? So that's why for us, it was a good model system. And this is the same cell type grown at high density and low density in glutamine. They're happy, they grow. No glutamine, they don't grow. Very simple, and that happens at low or high density. So now we're going to add, instead of glutamine, this dipeptide, and this is what you see, right? So at high density, they're super happy. They can grow on it almost as if it would be glutamine, but at low density, they're completely unable to use it. It's a movie of that, right? You can see these two populations at high density, they're almost the same derivative here, but you see the in dipeptides, even if actually they start a little higher, they collapse after 2 days. As I said, you can do this with dipeptides and tetrapeptides. These are the reviewers asking to do all these things and all different combinations. So basically, no matter what kind of oligopeptide we try, it's the same idea. Dense populations can use them completely as an alternative source of amino acids. Low population cannot. So how do oligopeptides get into the cell? And I'm going to just, again, save you years of work to say that they do not get into the cell. So what happens is that the cells secrete peptidases, cleave the oligopeptides extracellularly, and then incorporate the amino acids that come from that collective digestion. And that's how it becomes a cooperative process. So in the next few slides, I'm going to try to show you what's the peptidase that does this and how does it affect tumor growth. So the first thing we want to do, and if you ask me what I do not want to work as a postdoc or in my lab, was proteases and peptidases. Sometimes data takes you to wherever you want. Now, the good thing of working with peptidases is that it's been, I think it's one of the oldest field in molecular and cell biology. So there is tons of inhibitors where you can target different families of proteases. And we basically did a very simple screen. So a bunch of inhibitors targeting different types of proteases, see which ones would inhibit the growth on dipeptides in dense cells, but not on amino acids, right? So that allows us to really quickly remove anything that was toxic. And we found this one hit, which is called bestattin. It's an aminopeptidase inhibitor. And this is the type of result that we get. So these are the cells growing on their own with glutamine, super happy. You remove glutamine, they do not grow. You can rescue because they are at a fairly high density with the dipeptide. But if you add bestattin, they cannot grow anymore. And now we can rescue that just by simply adding back glutamine, where now they don't need the peptidases because they have the free amino acids. So there's nothing toxic of bestattin. And if you work in any cell type, I challenge you to test this because we haven't found a single cell type where it doesn't work this way. You can see there is some quantitative differences across different cell types. And this is just a subset, but qualitatively, we see the same thing, right? So we think we found the type of protease. Now, the problem of proteases is that there's a ton of them. So there's like about 50 human aminopeptidases. So it took us about 3 years. Now that's great. During COVID, fun to narrow down. And, we were like constantly panicking. What if it's like a redundancy of 3 of them, which would be very biologically feasible, but not the kind of thing editors or reviewers want. So we were like traumatized. But we ended up doing, after many fails of different approaches, partly because you cannot do pooled screens here, right? Just a quick side note, because it's a non-cell autonomous process. So if you have a subpopulation that is knockout for the right protease, but the rest of the plate, it's knockout for different ones, they will rescue the growth of the knockout, right? So that was one of our limitations. So what we did was take conditioned media, do proteomics, see which peptidases are present of this particular family. We found 16 of them. We did some sort of small combinatorial deletions, and we found a single one that was completely required for this process. So maybe I'll just show you here. It's called CNDP2. It's usually thought to be a cytosolic peptidase, but actually we do detect it in conditioned media. If you knock it out, cells completely lose the ability to grow in dipeptides, and you can rescue that by re-expressing it. So that was super cool. And this is, I'm just showing you one particular cell type, but we've tested at least in 10. So that works. And this is when things started getting exciting. So you can actually dramatically reduce tumor growth by knocking out these enzyme. And I'm, again, sparing you a lot of detail, but what we do is that we place mice in a nutrient, in a amino acid reduced diet. So there is a diet that has no glycine and no serine. So that's really forcing the tumor to, at their maximum capacity of their own making of amino acids. So they're really sensitive to any perturbation of the amino acid metabolism. And in that condition, you can almost get rid of, like 75% of the mice do not develop tumors in this system just by knocking out the peptidase, right? So the idea is, if you have plenty of amino acids, and that's probably what happens with the rest of the cells, if you have the amino acid that circulation is delivered to you, this process becomes completely redundant. But in ischemia, where the tumor is not well perfused, or in these conditions that we're even tuning down amino acids even more, this becomes the backup mechanism. And in what we're doing is sort of ruining the backup mechanism. We did a little bit more of a clinical tumor. So this is for like the cancer connoisseur. So this is an orthotopic tumor model, and we're using a very common patient combination of mutations. And in this case, we're not using the knockouts. We're just treating with the inhibitor, and we also see a fairly significant reduction on tumor burden. So we submitted this to like a British pamphlet, and it's been like 2 years of nightmares. So hopefully that ends soon. But one of the main concerns has been if they secrete these peptides, which to me is yes, because we detect it outside. So, but that's going to quickly show you one cool experiment. I think it hopefully now convinced the reviewers. But so first of all, we see the protein in plasma, and in this case, it's bronchial alveolar fluid. So it's basically lung interstitial fluid from tumor-bearing mice. So we see it. There is tons of evidence in patients. If you look into random studies of proteomics of plasma proteins, you see it. So, but is it functional, or is the function extracellular? So what we did is we took bestattin, this inhibitor, aminopeptidase inhibitor, and we added some long or relatively long pegylated chains. And the idea was that we would make this small molecule that is very cell permeable, cell impermeable, because the pegs are very charged and long and big. So hopefully, if this inhibitor still works, it means it's working extracellularly. So this is cell culture. So this is cells growing in high density again on amino acids or in these dipeptides. If you add bestattin, no problem with if they're working growing on glutamine. But if you are using dipeptides, they cannot grow. And then we did all these different PEG variants, and it works exactly the same. So the compound at least didn't lose its activity. And then we pick one of them and synthesize a lot more, spend a lot of money doing that. And we tested in vivo. And again, we see the same thing. So the effects of tumor growth of this extracellular bestattin are very similar to the regular bestattin, really suggesting that targeting extracellular peptidases is important for this particular process. So this is the model we're working on. So basically, in low density, if your source of amino acids happen to be dipeptides, there is not enough cells to do the cleavage. And I'm pretty sure someone's going to ask why it doesn't work in low density. So I can get more into that. But then when the cell density is higher, they accumulate enough proteases and degrade enough oligopeptides so they can grow. And it happens to be that this peptidase is called CNDP2. So just to end, I wanted to kind of go back a little bit to the sort of big picture problem. And despite I like cooperation very much. And I think maybe even us as humans, as a society, could benefit a little bit more of cooperation. You can see here, it doesn't exist on their own, right? Like, so there's always cooperation and competition, right? So here's some hyenas cooperating to chase away a lioness. But I'm sure as soon as the lioness leave, they're gonna start competing for scraps, right? So these two processes are not mutually exclusive. So what happens if you have now the CNDP2 knockout cells and the wild types, right? And what happens if you co-culture them, for example? So it kind of happens what you would expect from those typical sociobiology experiments. So this is the growth of wild type cells in dipeptides. This is the knockout, so they cannot really grow. And then if you co-culture them, right, the wild types grow a little slower, basically because the knockouts are using some of their glutamine, but now the knockouts really grow at the same speed, right? So that's pretty cool. So we decided to do some long-term cultures and see what we would expect is that either they will coexist forever or that maybe the cheaters would actually sort of expand a little bit. And I guess I don't have the data of the long-term experiments, but basically, if you do this over several passages, at the end, you just get the wild types. And that was confusing to us because, again, if you just see here, they're growing at exactly the same growth rate. And it seems that they are completely capable of taking advantage of the amino acids cleaved by the cooperative ones. So we went back to that old system that you all forgot about I introduced you at the beginning, where we can have these gradients of nutrients, right? And we wondered what happened with the coexistence there when you have progressively lower levels of nutrients. And hopefully you can see that when conditions are fine, cooperation is maybe less important. So you see that both wild types and knockouts coexist here very well. But when condition becomes rougher and amino acids become more limiting, it's only the wild types that can survive. And the reason why basically this happens, and we've shown this, is that basically the cooperation becomes really, really local. So if 4 cells away are not cooperating, then you don't get the benefit. And that's how you establish this. And actually this happens in vivo, so we can co-inject these wild types and knockout cells with different fluorophores. And what you see is that after 30 days, the knockouts are completely absent. And when you look at tumor section, right, so you see this section here, this should be 50-50. That's what we injected at the beginning. There's mostly wild type cells. There's a few pockets of knockouts. And it does seem that the knockouts are actually localized in regions with high nutrients. So it does seem that even in the tumors, in the few regions where perfusion is abundant and nutrient levels are well maintained, you can get away without cooperation. But as soon conditions become rougher, it's only the cooperative ones that survive. So with that, just wanted to credit Natalia and Logan from the lab. They're the ones working on the biochemical version of these gradients, and we're working on a paper we hopefully submitting this month or next month. And then Gisem and Septiembre are the ones working on the cooperation project that we just resubmitted our 3rd round. Hopefully they accepted. And yeah, I'm going to start submitting papers only to bioRxiv, the only journal that always accept my papers. And then, yeah, just thank the generous support from many institutions, and happy to take any questions. Yep.
Yeah, so for the cheaters versus cooperative, at the end, you mentioned that the ones that have the knockout polypeptide cleavage enzyme do not succeed; they die out. How are you able to differentiate between? You said you looked at this over a long time. How are you able to differentiate this between you just passing them out because you're growing slower versus they actually die?
Yeah, so the question is really how, when we have these co-cultures between cooperators and non-cooperators, how can we distinguish if it's they are dying out because of a lack of cooperation or if there is differences in growth rate? So That's a good question. So one of the things we do is like our control is the same two cell types growing on glutamine, so growing in single amino acid. So that gives you a baseline. We've done as much as we can do. We don't see any difference in their growth rates in glutamine and in co-culture. If they are well mixed, they also grow exactly at the same rate when they are co-cultured together. What we think, and we did some back of the envelope calculations of the diffusion of glutamine and of the peptidase, is that basically it's almost like if you're 1 or 2 cells up, if your closest cooperation is 2 or 3 cells apart, then you're no longer getting the benefit. And you have to think these cells are adherent, right? So what happens is that if you have 2 single cells, when it's wild type, one's knockout, the wild type will divide, and then the 2 daughter cells are going to end up basically right next to each other. So they're going to cooperate really easily. Now, if you have a knockout cells that is right next to them, it's going to benefit from that. But then when that divides, one of the daughters is going to be now a little bit further away from these cooperative clusters. And if you expand that over time, you get these ever-expanding patches of cooperators. And the non-cooperators are basically, they can only survive basically on the edges of the cooperators. And if they step out a little bit into the wild side, they just can't do it. Should I read them? All right, so so Diana asked, can you clarify what is a cheater again? So another contention point in the review process, so we are now calling them non-cooperators. Traditionally in sort of social biology, where you have, for example, in bacteria cooperation, a cheater is a cell that can take advantage of the cooperation, but doesn't pay the cost of cooperating, right? So you could imagine in bacteria, if you have a non-cell autonomous antibiotic resistant gene, you could imagine that cells that do not produce that can still be protected if their neighbors produce it, but they don't have to do the sort of metabolic cost of making that antibiotic resistance. The reason why we don't call them cheater in our system, even if we did in the first version, is that we don't really see, or we haven't been able to measure a significant cost of this cooperation. And to be fair, I cannot imagine making or not making one single amino peptidase really has any sort of effect on the cell growth of a cell, right? They're making thousands of proteins, so so we don't see a cost. So we are calling them non-cooperator in the sense that they can still take advantage of the cleavage produced by these the ones that secrete the peptidase, but it's not that they're skipping a cost. I hope that makes sense. So what are we calling non-cooperation? As a short answer is the CNDP2 knockout cells. Yanis was very generous in posting the website of my lab. So thank you for the advertising. And then Joao asked, thank you very much. You're welcome. In the case of ischemic environment, where the allele threshold is higher, is the carrying capacity lower because of competition? That's a really cool question. I think so. So I don't know if it would be lower because of competition, but the carrying capacity could be space, could be oxygen, could be nutrients. So, right, in a typical, in the blood, let's say the levels of glucose are 5 millimolar. So if the growth would be limited by the amount of glucose available, that would be your carrying capacity. As you go into the gradient, if the actual levels of glucose are lower, you could imagine that the maximum number of cells that you can have will be limited, because you cannot get, even if there would be no competition, you cannot get the same amount of cells because the total amount of glucose will be lower, if that makes sense, right? So it would be like if you have two vials of cells where there is no competition, because you have a low enough density that there's no competition and there's no problem of cooperation, the cells with 5 millimolar glucose are going to grow more than with 1 millimolar. That makes sense. But yeah, we haven't really thought in that way or measured it, but I think that's a really good point. So yes, I think as you go into ischemic regions, you have a lower carrying capacity. Yeah. And then Diana says, okay, thank you. So we completed the round. Yeah. Everything is for them. Thank you. Mhm. Exactly. Yes. Yeah. Yeah. No, that's a great question. The question, what's your name again? So Sanket is asking whether we're planning to look at the same kind of interactions in vivo. And so we did that experiment, which is a little bit of a toy experiment of co-injecting the cooperative and non-cooperative cells and see how they evolve in the tumors. Maybe I went through that very quickly, but basically, if you do that in a tumor, the non-cooperators disappear fairly quickly. So that is one way to look at that in the tumor. One of the things we want to go forward now, one is to develop maybe potential therapeutic interventions, taking advantage of this process. So we want to now, especially that we have this pegylated form, we want to develop a better extracellular aminopeptidase inhibitor. So that is where I think we're going to go next on that story, maybe walking a little bit towards translation, but continuing with the idea of the cooperation and that sort of bigger picture. I want to not move away, but move forward or like beyond just CNDP2 and start thinking about how specific oncogenes or tumor suppressors may affect the sort of evolution of a tumor, right? So that's why I'm always so excited about this Tumor Dynamics Center, because you could imagine that, depending on the kind of mutations that the other clones have, having a particular mutation or not may have a very different outcome, right? So we would like to start looking at patient data and sort of all the sequencing data that is available. If you can combine it with, spatial omics and with what you guys are doing with these CRISPR model, pool screens in tumors, that would be really cool. With the idea of, maybe KRAS mutant P53 knockout tumor cell has a much tougher job growing if the rest of the tissue, it's also KRAS mutant than if it's wild type, right? So we're trying to see if there is sort of non-intuitive predictions that you could do that could indicate really sort of patient-relevant sort of cooperative or competitive interactions, and then try to model these in our system, right? And then again, also these different oncogenes may be very different, beneficial or not, according where in this gradient of nutrients you are. So model it in the yeah. If you have any nice okay, that sounds wonderful. So my goal here is to get help from you guys to look at this omics data and get some mRNA and guides across tissues and in our system, and then also modeling, right? So then that would be the cherry. So yeah, we at the moment, I think my carrying capacity is the number of people, right? Like I always come with, like, let's look at this new cell type, and they're like, oh my God, right? So, but you are exactly hitting the kind of things we are interested in, right? So in our system, and my interest is, you could probably tell, it's like really the biology of these cells and the tumor, but we do want to keep some link and loop with what's clinically relevant, right? So that's why we are also really interested in seeing what can we see in patient databases and how we can try to model or explain some of that heterogeneity with our system, right? Have there been, so Yanis again is asking, have there been any experiments with human cells, and what differences from mice can we anticipate? So in terms of our cell culture experiments, we've done a lot with human cells and mouse cells, and we don't see big differences. So everything I show you for both projects happens with humans and murine cells. In terms of in vivo experiments, obviously only mouse, and we've looked at some databases on CNDP2 alterations in tumors. It's a little bit difficult because what we would be looking for are that tumors do not lose that protein, and indeed, it's fairly rare that it's absent or mutated. So it's harder to predict on that end. But that's why I think, going to the spirit of the question, why we want to get now, hopefully this approved, so accepted, so I can start thinking on something else. And that something else is probably trying to go to these big databases and hopefully also some spatial omics, and see whether we can extrapolate these the ideas of these interactions to other genes and other mechanisms. I have all day. I'm here all day for you. Mm-hmm. Yeah.
And stuff like that. So how do you take that
Yeah, so that's a great question. So the question, if anyone is still in Zoom, is that how do we take into account cell death that presumably happens more in the more ischemic regions? So if you do an experiment that is long enough, it's really cool. You actually get first, for example, cells becoming hypoxic or ischemic, and then you start seeing the emergence of a necrotic core. So the very extreme cells die and disappear, and then before that, you have a sort of sublethal. In the kind of experiments we do for the T cells, for example, we don't see any evidence of cell death in that time scale. In fact, if you don't have T cells, for example, in the tumor cells, there is virtually no cell death. And when we add the T cells, there is also really not, like, that's what we're looking at. Actually, tumor cells are surviving. So it would depend on the time scale that you're looking at. Now, to the other, the interesting point I think that you're also trying to hint at is that what is the source of oligopeptides? I'm talking about this sort of, I'm a survivor cutting the plants with the machete, but there's also all this cell debris. And we think that can also be a big contribution in vivo for resources that then tumor cells scavenge. And we've been studying this in amino acids, but I don't see why there couldn't be a similar mechanism for lipids or for sugars. And that may or may not be cooperative, but it seems that there is a lot of nutrients available to tumors because of all that cell debris. We don't think, for example, that was another discussion with this reviewer, like one of their ideas that maybe CNDP2 is not secreted, is actually just leaking out from cells that are dying. And we've shown that this happens even if viability is super high. We don't see other proteins that you would see if there is loss of cell viability. So in those kind of experiments, we don't think it's particularly relevant, but I absolutely think that cell debris and cell death, it's a source of biomass that tumors would be, to me, would be evolutionary and feasible that they're not taking advantage of that. Uh-huh. Yeah, like in the ischemic regions. Yeah, no, they're fine. So we have an experiment. You have them in between these glasses and they don't kill. And then we can break the glass, literally, like, well, a little bit. You got to be careful because, but you can remove the glass. And then the next day they killed everybody. So they're not only alive, they can recover the ability to kill. Actually, what we think is that they never lost the ability to kill. It's just the tumor cells are hiding because they're not exposing MHC1. So if you literally break the glass and remove the gradients, by the next day, the tumor cells have MHC1 again. They're exposing antigens and the T cells kill them. Well, we haven't really looked at that. Like, if you look at the way, like, I don't know if you work with T cells, but basically when you have these killing assays, at least with microscopy, it really looks like a mess of cell death. So we haven't really